Hey everyone, good evening.
Good evening, Professor.
Hi, it's December now.
Good evening.
Good to see you guys. So I'm gonna go ahead and get started. I think I'm recording. Yep. Today we're gonna talk about a little bit of a special topic, but it's still within the realm of all of the technologies that we've been kind of talking about as we go through the semester. And then I want to do a trivia game with you guys. So I actually made a Kahoot trivia game to follow this afterwards. So after I kind of slog you through all of the information, then stick around to play the trivia. So tonight I want to focus on a topic that's dear to me because it's the area that I work in, and that is the topic of omics. And omics is kind of a broad term that refers to a detailed or comprehensive analysis of basically any biological system. When we say system, sometimes it sounds confusing, it sounds very mechanical. What we just mean is very broadly, it could be something as large as a whole organism, like a whole human, or it could be something like just a heart. So a system could be any variance in between that, right? So a whole entire organism, or maybe just a cell in an organism. Anything that works as a system in biology. And the area that this name came from, omics, is from genomes. So you probably are familiar with the term, at least genomics. And that's because this is where we coined the term. When they did the sequencing of the human genome, we ushered in this whole area of systems biology, or again, just where we're focusing on a whole comprehensive analysis of a system. And that's where the omics technology came from. But we can now, and we do use the word omics away from just genes, we can use it to just generally talk about the collective use of anything. So for example, anything that ends in omics is just the study in biology of the whole system of that thing. So genomics is all of the genes. There's also metabolomics or proteomics. So again, it just is any of those fields respectively where you're studying the whole data of that particular system or organism. So we're going to talk about these four main omics. There's actually a lot of omics. They start getting very specific. There's even things called like nutritional omics or toxicomics. They start getting very, very, very specific. And now that this field is out there, there's more and more and more terminology that's coming with it. But these are the four main omics that you will see, and most of the omics are related to these. And each of them is related to a particular kind of area of that central dogma that we talked about in biology. So there's genomics, again, which is the study of the complete set of genes or DNA in an organism. There is transcriptomics, which again is going to be the study of a complete set of RNA in any system. And here it's not RNAomics, right? Genomics is genomics. Remember that this process of DNA to RNA is called transcription, and that's where we get that transcriptomics. The study of all of the proteins in a system would be called proteomics. And then the very last one, and this is the one probably people are not as familiar with, is the study of all small molecules in a system. So this can be broadly classified as literally anything that is a small molecule and exists in a biological system. So anything that's not a protein, that's not RNA, and that's not a gene, and it's small. It could be a neurotransmitter, it could be a sugar, it could be anything. That all falls under the umbrella of metabolomics. So we're studying metabolites. So first, let's look at genomics. Again, genomics is the study of all the genome of an organism, and the genome is an organism's complete set of DNA. That's all of its genes. So this is in contrast to something like genetics. If you ever hear the term genetics, this is where we're looking at the study of individual genes. So we might be focusing on one gene and understanding that gene and their roles in inheritance. So if you've taken a genetics class, you probably have talked about how a particular gene influences something like eye color, and how that is related in your genetics, right? How that can be passed down. Again, genomics, in contrast, is where you're taking an organism and you're profiling all of the genes that it has, and you're looking at those genes. The areas that the topics this aims to get at in genomics is you get to characterize and quantify an organism's genes. And it's going to give you information about those genes interrelations and the influence on whatever organism you're looking at. So if you're looking at a human, you might classify all of its genes and then say, oh, this gene looks different than another person's gene. And so we know that this gene influences certain diseases. So generally, in order to compare DNA in genomics, we have to already have a genome map, right? We have to have an established set of DNA that we know exists to then compare the DNA that we collect from a person or from an animal or from a plant to. So this just kind of highlights one of the importances of the Human Genome Project. The whole thing that sparked genomics was we first mapped out the human genome. We know all of the genes that exist in their potentiality in a human, and then we can compare any of the DNA that we collect from a human to that map. So genomics needs to have this kind of already established roadmap in place for you to then compare to. One of the main uses of genomics is to kind of understand diversity at a whole genome level. And this is going to give you insight into things like traits and disease susceptibility. Generally, you're comparing. So you're taking a group of people who maybe have a disease, for example, and you're profiling all of their genes, right? So you're going to collect all the genes from all the people who have a disease. And then you might collect all the genes from people who don't have a disease. And what you would then do is compare. You'd say, I have all the genes from these people that have a disease and all the genes from the people that don't have a disease. What is the difference between them? And you would find maybe, oh, hey, there's a genomic variant between the people that have a disease and the people that don't have the disease. And this is a way that we can tell genomic susceptibility, for example, the potential susceptibility to get a disease if you have a particular variant. This is also how we can kind of highlight specific traits, right? Phenotypic traits that exist, for example, things that we express phenotypically would also be able to be categorized using genomics if you did comparison of people who have or do not have a trait. Now, genomics is static. This is the one thing about it. You're just getting that person's DNA. You're not, for example, the person might not have a disease, but they might have the potential to get a disease. All you have is their set of DNA. You don't know what's going on phenotypically, what's being expressed in their body. For example, just having someone's DNA doesn't necessarily mean that they have cancer, right? You can't tell that from their DNA. You might be able to tell the potential that they are more likely to get cancer because they have a trait that's connected to people who usually have cancer, but you can't definitely for sure say just from their genome, from their DNA, that they at this moment have cancer or do not have cancer. So I want to talk about technologies that you would use to analyze the genome. And I want you guys to think about when we talked about DNA and nucleotides and the analysis that you would do to figure out um, a strand of DNA or to analyze. Remember, here we have to analyze a whole set of genes of a person. What kind of technology do you think you would use? So I want you guys to respond in poll EV and think about technologies that could be used to get the whole set of genes. Again, think back to technologies we have talked about in class. All right, I'm seeing some coming in. People are saying PCR, microarray, amplifying and sequencing. So some of these, like genome sequencing, I'm asking more specifically what exact technology you would use. So yes, we are sequencing the genome, but what do we do? What do we use to do that? So again, think about what the technologies we talked about that actually tell you what the DNA strand is. Ooh, someone put Western blot. Remember, Western blots are for proteins, not for DNA. Okay, so the technologies you would use for sequencing DNA. Remember, we have a sample of a person's whole DNA, and we need to sequence it. We need to see what their DNA strand is so that we can then compare it to potential other DNA strands. So one thing that is done, this isn't the way that you sequence it, but something that can be done for sequencing, if you guys remember, was gel electrophoresis. Remember, we looked at those gels and you could tell the sequence of a short set of DNA, right? You could say like it's A, T, C. You guys even had a homework problem where you had a gel and you had to figure out the sequence of a strand of DNA. Now, gel electrophoresis is not usually used for genomics because it's only really good for a shorter sequence, like something 100 to 1,000 base pairs long. And remember, for genomics, we're literally trying to sequence the whole set of someone's genes, all of their genes. So the two things that are utilized the most are what are called shotgun sequencing and high throughput sequencing. Shotgun sequencing uses something you guys are already familiar with, and that is the Sanger method, which again acts kind of similar in the type of information you get out when you read your gel. But remember, it's going to be using either a radio label or fluorescence to read out each of those base pairs that come. So you could get A, T, T, whatever, etc. Another type of sequencing technology that is used is called high throughput, and this uses, in particular, something called Illumina dye sequencing. Now, this is not one of the areas that we particularly covered, but I wanted to highlight this is one of the newer types of technologies that's being utilized for genomics. So again, the main one that you guys have talked about in how to sequence DNA is this Sanger method. And for shotgun sequencing, they're doing it at a much larger scale, but it's still the same idea, the same principle. Can any of you guys tell me why you wouldn't use PCR? A lot of you answered PCR. Does anyone know why would PCR not be good for sequencing the whole genome?
Because it can just amplify a segment of DNA, not the
That's
whole.
That's right, Hakima. Remember, the purpose of PCR is it just amplifies a segment of DNA. So you put a primer in for PCR, and it just tells you if that segment of DNA is there. It's not going to give you the sequence of the DNA. In fact, you already have to know a little portion of the sequence to just amplify that one gene per se. So if we want to collect, we want to get the whole genome, we want to know the whole sequence of DNA, we have to actually sequence the DNA using these sequencing technologies. Okay, so let's now move to transcriptomics, which is, again, looking at the study of transcripts or RNA. So here we're studying all of the RNA molecules. That includes mRNA, messenger RNA, ribosomal RNA, transfer RNA, and other non-coding RNAs. There's actually lots of types of RNAs. The main one that people focus on is mRNA. Oops, remember, messenger RNA is telling you, it's the one sending the message, right? It is the one that is telling the body what proteins to make. It's the messenger that came from DNA to make proteins. So if you were to do a transcriptomic study, you could collect RNA from either an entire organism. This would be a little hard for example, if I wanted to get all of the RNA out of a human, that would be hard because I literally would have to extract every single piece of RNA out of one person, right? I'm not going to like squeeze out or crush up a person and get all of their RNA. So usually we're looking at the RNA in a particular tissue. So you might have a sample of a tissue or a biofluid. I might just be interested in what is the RNA levels that are in the blood, for example, that are circulating through an organism. You could get all of the RNA out of a small organism. For example, if you're interested in the transcriptomics of just a cell, and you had a cell population, you could then extract all the RNA out of those cells. You could do that for something like a cell culture, if you were looking at cancer cells or liver cells or anything like that. Or you could extract it out of a whole cell organism, like a bacteria cell, if you're interested in what was going on in a bacteria. But when we're talking about large organisms, generally, because the RNA levels are different all over an organism, right? The messages that my liver are sending are different than the messages that my brain are sending, and they're different than the messages that are circulating in my blood. So usually we're just focusing on one of those areas, a tissue or a biofluid, and getting information about the RNA there. Now, transcriptomics, something that's nice about it that is different from genomics is it's going to give you a snapshot in time of all of the transcripts in a cell. So it's dynamic, right? It's giving you a whole account of all of the processes that are active or dormant at that time, right? Remember, your DNA is just the main blueprint of your body. We don't know which genes are turned on or turned off on that DNA segment in a cell. Your liver cell might be upregulating a particular gene, while another cell in your heart is not upregulating that gene. So transcriptomics can tell you this. If you're looking at a particular area, you can get the gene expression, you can get gene expression information from the RNA by sampling your tissue or biofluid. Now, I want to be clear here that although you are measuring the RNA, for example, the messenger RNA, that doesn't necessarily correlate to how much protein is ultimately made or is currently made. So just because you have sent out a bunch of messenger RNA in your heart to upregulate a particular protein, it doesn't necessarily mean that all of that protein is then going to be made. This is something that we actually know now. We used to try to correlate them. We used to say, oh, however much messenger RNA is there, that's how much protein you're going to make. So they're connected. While generally they're related, they don't always correlate. And I want to just be careful to make that clear here. Again, transcriptomics is useful if you're doing a comparison analysis. For example, healthy cells versus some disease cells or cancer cells or any type of pathological cells, maybe cells that are treated with a particular toxic substance. So you're comparing two things together, and you're going to see how the gene expression is altered between those cells. So usually you will collect all of the RNA and then you tag it differently depending on which cell it's coming from, so you know which cell it's coming from with a fluorescent probe. So I'm going to clear this. And again, I want to ask you guys about technologies you think you would use, but to measure RNA. So here we're talking about comparing the levels of RNA from a tissue or a cell or a biofluid. What kind of technology would you potentially use here, if you know? And remember, we need to analyze all of the RNA that is coming from a cell. Remember, omics is not just looking at one particular RNA or gene, it's looking at all of that genes or RNA or proteins that are in a sample. So you have to analyze a lot. Ooh, someone's still answering Western blot. Remember, Western blots are for proteins. Took it down. Okay, so this one is not as straightforward because we haven't directly talked about either of these two technologies, but one of them is similar. Someone put the high throughput sequencing. One of them is similar to how you would sequence DNA because RNA is similar to DNA in its structure. One of the main ways you can measure transcriptomics is by doing something called a microarray. I kind of showed you a little bit of what a microarray is here. So if you collected all of your RNA segments and then you tagged them, what you can do is take a chip that has a complementary RNA segment on each well. And what you do is you put all of your sample into each well and you see if any RNA binds. If it binds to the complementary segment, it will light up. So what this tells you when you read this chip is, you know, for example, which RNA segment was in this well. And you can see, for example, here that neither sample bound. So neither sample had RNA that was transcribed for this particular gene, this RNA segment. This next well over, what you can see is it is a red color. So that means that only the pathological cell or the disease cell in this case, which is a red probe, expresses this RNA. Now, if it's present in both, for example, if they both equally express it, then it would show up as yellow. And these chips are huge and you do a vast amount of them because every well has to have a different RNA segment in it to see if it binds to that. And you just go through all through the chip and see what genes are expressed and in what level, and you can get an idea of the type of RNA expression that is happening in your disease versus your control. Now, another type of technology I'll talk a little bit about is called RNASeq. And this one is accomplished by essentially reverse transcribing RNA back into DNA and then sequencing it. And it does this all very fast, so it's going to sequence each of the codes. And you would use something similar to like the sequencing we talked about for DNA to do this. So the first thing you have to do is make it into short segments of DNA. So you take your RNA, you reverse it back into DNA, and then you sequence that DNA. These two areas are very hot right now, RNA-seq and microarray. RNA-seq is the newest area of technology in transcriptomics. And I just wanted to give a little bit of comparison between the two of them because microarray and RNA-seq both have some pros and cons when you compare doing one or the other, why you would do one or the other. For example, if you look at how fast they occur, microarray are much faster. It takes 1 to 2 days per experiment to get all of the data from a microarray versus an RNA-seq, which can take up to a week to do all the full sequencing. If you looked at the amount of sample you need, RNA-seq wins out because you only need up to a nanogram of RNA from your sample versus a microgram for microRNA. RNA-seq is much more labor intensive because of the preparation and data analysis you have to do to read all of the sequences, whereas microarray is pretty low labor intensity. As you can see, you literally tag things and put them on a chip. The chip's pretty easy to read because it's just color-coded, whereas RNA-seq, you have to actually get all the information from the sequence. Now, if you were to do an RNA-seq versus a microarray experiment, you don't really need to know anything going into an RNA sequencing experiment because you're sequencing everything. It's just going to give you all of the sequences out. You don't really need to know if they're useful or not. You can determine that later. When you do a microarray, one of the things about them is that they're limited to the chip size. You can imagine that having one of these chips that literally has millions of RNA probes on it would be very hard to make. So usually people limit the chips. They might only have a couple hundred or a couple thousand RNA sequences on a chip. And they'll kind of cater what RNA sequences are attached to the chip based on what they think might already be there. So for example, I'm not going to put RNA sequences that I know come from a banana if I'm analyzing a set of RNA from a human, right? Or even if I was going to look at some RNA from a lung cell, I might not include RNA from brain or other tissues that I know are only in those tissues on my microarray chip because why bother? It's just going to be a bunch of empty things. So you kind of have to know a little bit about what you're looking at before you do a microarray versus an RNA-seq. Now, conversely, the thing about RNA-seq that is good is if there's something there that we don't already know about, then it will find it because it's going to sequence everything. Now, then there's just some differences in how sensitive and the range that they have. RNA-seq usually also wins out in this sense because the sequencing is going to be more sensitive than in a microarray. Okay, so that's it for technologies on transcriptomics. Let's now look at proteomics, which is, of course, the study of all of the proteins in a system. It can also start to look at their structure and function. That's kind of a little other subsection where we start getting more detailed in not only what proteins are there, but how they work. So again, the proteome, the whole set of proteins, is all the proteins that's produced or if they are modified by an organism or again by just a system. It confirms the presence of the protein and it also provides measure or quantity of the protein that is present. So that's another nice thing about proteomics is it's not just giving you a yes or no if it was there, it's giving you how much protein is there. Just like transcriptomics, similarly, you can collect these from any organism, tissue, or a biofluid like urine or saliva or your blood. Again, it's going to be easier to focus when you're looking at a large organism on things like just the proteins in a particular tissue, just the proteins that are in the blood, etc. Whereas if you look at just a cell or just a small organism, you could collect the proteins from the whole organism and look at what's happening. Like transcriptomics, proteomics is also dynamic, right? It gives you a snapshot into what proteins are being expressed right at that moment when you are collecting the sample. So because of this, it's more complicated than something like genomics, because again, the genome is constant, whereas the proteome is going to differ from cell to cell and from time to time, right? Again, as I said before with the transcriptomics, your liver cell is going to be utilizing different enzymes and different proteins than your heart cell is. And not only will different tissues use different proteins, but tissues will use different amounts of a protein at a different time. For example, your pancreas might upregulate or downregulate your production of insulin based on how much sugar is around. So we know that the pancreas will make insulin, but depending on the time that you sample cells from a pancreas, the insulin levels might be higher or lower. So again, it's a snapshot at that time and it's very dynamic. So similar to transcriptomics, one of the ways that we use proteomics is in comparison type of studies. Usually it's going to help us identify main proteins in a particular sample, and proteins that are expressed differently in related samples. So again, an example here would be disease versus healthy tissue, how those proteins are being differently regulated. A lot of studies will, for example, look at normal cells versus cancer cells. Remember again that these cells have the same genetic code. They have the same blueprints of DNA, but the amount of proteins that they're making, what they're expressing, what they're doing is very different from each other. So something had to happen to make them different, and we want to kind of look at what is different about them first to figure out what is going on. Proteins that are found in, for example, diseased samples can be very good because they can act as a drug target or as a diagnostic marker. So for example, here we're looking at a normal cell versus a cancer cell and a particular protein called the HER2 protein. Both of these cells make the HER2 protein. They have the genetic code to make it, and they both make it. But in cancer cells, it was found that this protein is amplified, meaning there is much more of it. And you could figure this out in a proteomics study, right? You could say, here's my healthy tissue, here's my cancer tissue. Hey, this HER2 protein is way more elevated in cancer cell. This can act as a biomarker. So if we were to collect information from a patient and they had elevated HER2 protein, we would know then that they potentially have, it's potentially related to this type of cancer. Also, we can use these as drug targets. Maybe this particular protein helps a disease function or helps the disease do what it does. So you can target that protein that is special or upregulated in a disease to take down the particular disease. We also know that while profiling proteins, we could potentially figure out functional relations. So proteins that are expressed similarly at similar levels might be functionally related to each other. So you could, instead of figuring out a biomarker for disease, you could figure out how a particular mechanism of protein works. You could say, hey, I noticed that when I eat glucose, both of these proteins get elevated. So they must be both related functionally to glucose. So again, I'm going to give you guys a chance. We've definitely talked a lot about proteins. So I want you to think about what are technologies we could use. I want to profile all the proteins in a sample, not just one protein. I want to look at all the proteins in, for example, a blood sample. What kind of technology do you think we would use? So again, think about types of technologies we use to specifically identify proteins. All right, we're getting some SDS page. Remember that SDS page is a particular type of gel electrophoresis. So we wouldn't just say SDS. SDS is a chemical that gets added to the gel that's helpful for proteins. Just to clarify for you guys. So the whole name is SDS page. You could also just generally say gel electrophoresis because a page or an SDS page is a gel electrophoresis. Someone also said PCR. So we just talked about what PCR does. It just amplifies a segment of DNA. So PCR is not really that useful for omics technologies because it's just looking at a segment of DNA, but it's very good at doing that one thing. All right, so we're getting some mass spec, capillary electrophoresis mass spec, Edmond degradation. So remember, we want to analyze proteins here, and we want to be able to classify all of the proteins in a sample, not just one protein. Okay, so generally, proteins can be detected using, I'm going to say, general antibody technology. So there's a lot of antibody technologies. These can sometimes be called immunoassays or antibody arrays. They act very similar to that microarray I showed you, where they're going to be having a whole set with each well has a different antibody. Every antibody is good for one protein, and it will tell you whether or not that protein is there based on if the well turns color, if it attaches. The other main technology that we use is mass spectrometry, which some of you guys said. Now, I do want to clarify here because some of you guys said this isn't a detection technique that's going to be good for like detecting specific proteins, but when you have a complex sample, you do usually need to separate it out first before we analyze it, especially for mass spec. So we will separate all of the proteins away from each other and then do analysis to see what protein they are using mass spectrometry. And one of the ways that we do this, the main way we do this, is by doing a gel. Sometimes it will be a one-dimensional or a two-dimensional gel to help separate the proteins. Now, again, if you guys look at this, this is a two-dimensional gel that you're seeing here. It's very hard to tell exactly. Like, I couldn't take this gel and say, that protein is IHC1 protein, or etc. It's very, very, very complicated on a gel that has so many proteins to tell what every single protein is. So I want to remind you guys that some of these technologies that are really good when you just want to look for one protein or maybe just a couple proteins don't work very well when you're literally looking at hundreds or thousands of proteins, because it's very hard to separate them out. So you need to have something, a technology that's very specific and that you can do on a large scale. So again, this is something like an antibody array would look like. Here you can see these are two different antibody arrays. One of them is using these A431 cells. So these are the normal cells. And then these are the cells that have been amplified in some way. In this case, one of their proteins have been altered. So they're looking at how the proteins respond when you look at the normal cells versus this situation of an amplification. Similarly, down here, same thing, we have our normal cells. These are called HT29 cells. All of the proteins that they're expressing on this antibody chip. And then what happens when we give them UV light. So these are cells that have been exposed to UV light, same cells, but just exposed to UV light. And we're going to compare. You can see, for example, these two proteins got elevated. So this is actually a pretty small antibody chip array. You can have them larger. Remember, if you really want to look at hundreds of proteins and what's happening with them, you'd have to have a very big chip. So one of the limitations with the antibody chips is, again, just like with the microarray chips, you have to have a huge chip that literally has, you have to have an antibody for every single protein on that chip. So they can get very costly and they can just be very hard to make because you need to make sure you know every single antibody that goes to its corresponding protein. So if you have a protein that, for example, we didn't already know about, if there is a protein that's new, a protein that's specific to only cancer cells that doesn't get made in normal cells, then your antibody chip might not pick it up. It probably won't pick it up because we didn't already know to have an antibody for that new protein that nobody knew about. So the best type of technique in this case would be mass spectrometry. It's not as accurate. So antibody tests are very accurate. They will definitely bind the protein. But mass spec is going to let you look at all of the proteins. It's going to give you all the information. So it's similar to when we do DNA. It's like doing the whole sequencing of the DNA or the RNA. So like RNASeq. In these types of experiments, again, you just are extracting your proteins. There are two types of mass spectrometry, two types of analysis you can do. They're called bottom up and top down. I won't get into exactly what you do there, but generally just know there's the two different types, and they are going to take some type of spectra and then give you all of the proteins that were in a sample from that spectra. Okay, the last area I'm going to talk about is, again, that area that kind of encompasses everything else, which is metabolomics. So this technically is the study of the chemical processes involving metabolites. Here we're going to identify these unique chemicals, sometimes we call them fingerprints, that are specific to a process of a cell and their profiles. To clarify the term metabolite, I'm going to use this term. Again, it's just used for any small molecule. They can be any type of biomolecule. They can have various functions. They could be amino acids, so singular amino acids that are not in a protein. They could be singular nucleotides or nucleotides that are small and not involved in a big piece of DNA, something like ATP, for example, which is just one nucleotide with the 3 phosphates on it. They could be lipids. They could be semi-complex lipids, but still just one lipid molecule by itself. They could be sugars. Again, we're talking about singular small molecule sugars, not the complex things like glycogen. Or they could be various other organic molecules. So some examples that you guys probably know about that don't fit into these classifications of amino acids, nucleotides, lipids, and carbohydrates. Think about neurotransmitters, serotonin, for example. Where does serotonin fit in here? Or GABA, for example. These are small molecules that we definitely use and that are utilized by various tissues in our body, but they don't really fit into these specific categories, right? They're just a small molecule that we use. So again, anything that is a small molecule that's used in the process in our body gets put under this umbrella of metabolites. Now, I will say, because lipids are so diverse, and we actually just spent a whole chapter talking about the diversity of lipids, there's many vitamins that are lipids, there's so many molecules that are lipids, there's actually a subset of metabolomics that's called lipidomics. So this is an area of metabolomics where we just look at organic molecules or lipids. Now, metabolomics, if you're doing a study on metabolomics, just like what I said with the transcriptomics and proteomics, you can collect the whole set of metabolites, the whole set of small molecules from any organism. Again, usually a small organism like a cell. You could collect them from a tissue, but generally, typically, we're going to be looking at biofluids when we study metabolomics. So we're usually interested in the small molecules that are circulating a system. So things like, again, blood, urine, serum, cerebral spinal fluid, that's the fluid that circulates your brain. Any of these biofluids could be used to study metabolomics. Now, metabolomics, one of the biggest pros for it, it is the most what we call phenotypical description of a system. And again, remember, if we think about that map of DNA makes RNA, makes protein, and proteins are going to be used to regulate or be regulated by metabolites, the small molecules. They are the thing that is going to be the exact snapshot of what is going on in the system at that moment. One way to think about it is that the genome can tell us what could happen. The transcriptome can tell us what appears to be happening. Proteome can tell us what makes it happen. But the metabolome is actually what's telling us what is happening or what has happened. Some of the barriers in metabolomics is that it is a lot of data. It's very computationally taxing. So when you think about genes and proteins, remember we talked a long time about what makes up genes and proteins, right? Genes are made up of nucleic acids, and there's only a subset of nucleic acids, right? When we do an analysis of a gene, we know that we only can look for an A, a T, a C, a G, or if you looked at RNA, a U, right? That's it. That's the only things that genes are made up of. So there's a finite subset of what genes could exist. Same thing with proteins, right? Proteins are made up of amino acids, and if we were to sequence and look at the protein, there's only so many combinations of amino acids that can exist in the world. It's a lot, but again, it's finite. There are thousands and thousands of different small molecules that exist in you as an organism or in all the other organisms. For example, plants utilize other small molecules. We're constantly finding new drugs or small molecules that came from different plants and different organisms all over the world. So the variety of metabolites is huge. There's thousands of them. They also are variable in their nature. What I mean by this is, again, genes are only made up of nucleic acids. Proteins are only made up of amino acids. Metabolites could be any structure. They could be a lipid. They could be a small amino acid. Their structural complexity is completely different from one another. Some of them might be very lipophilic. Some of them might be very hydrophilic. They're all very, very different. And that makes it very challenging to be able to analyze all of them at the same time. So because of this, we have to utilize a lot of machine learning and statistics to do comparative analysis. Also, we still don't know every metabolite from each organism. We know the whole genome in a human, but we don't know every single small molecule that humans utilize. We also don't just not know every metabolite. There could also be mapped out functions for metabolites we don't know. So we know that we use tyrosine, but we might not know every single pathway tyrosine is involved in. Again, I want to say that just like when I talked about proteins and proteomics, usually because these samples are so complex, again, you could take a sample of blood and it could have thousands and thousands of different metabolites in it just from one sample. So because it's very complex, usually we like to do what we call a separation technique first. So remember in proteomics, I talked about how you would maybe run a gel first and then do the analysis to figure out what each protein is. Similarly, we will do this with metabolomics. We'll do some type of chromatography, usually high performance liquid chromatography or HPLC or gas chromatography. Remember, chromatography are just separation techniques. So we'll separate everything out, and as it's being separated, it will then go into the analysis instrument, which is going to be either an NMR or a mass spec. And again, the reason that we use these two is because NMR can distinguish molecules individual of each other, any type of molecule, because it's just looking at its signature of nuclei, right? Similarly, mass spec, usually you can distinguish almost any molecule. Can anyone think about an instance that would be hard to separate molecules on a mass spec? What's a type of molecule that's hard to distinguish using its mass? You can put it in the comments. Again, think back to your basic chemistry. What's a type of molecule that is hard to distinguish based on its mass? So Babak puts volatile molecules. Volatile molecules, the mass isn't going to be the distinguishing factor, but for volatile molecules, we'll use gas chromatography because they like to be in a gas state, and they can still very easily be analyzed by mass spec. But again, I want you guys to think about a case where I'm doing an analysis, I'm looking at thousands of molecules. What might happen? Where would there be a case where I have two molecules that look the same in a mass spec? There's a name for this. So again, think about not enantiomers. Enantiomers physically are mirror images. So remember, mass spec is just measuring the mass and the charge of a particular compound. So Forrest got it. It's isomers. Remember that an isomer is something that has the same chemical formula, or excuse me, yeah, same chemical formula, and so thus has the same mass, right? But it might structurally look different. Mass specs are not telling you what something looks like. They're not telling you the actual shape of a compound. Mass specs literally just tell you the mass and the charge. So if compound A and compound B both have the exact same mass and the exact same charge as each other, so maybe they just have one thing rearranged, it's very, very, very hard, and you basically can't separate them on a mass spec. Mass specs they're bad actually for looking at things like enantiomers, because they can't tell the difference of two things that are identical except that they just have a flip in their spatial orientation. You cannot distinguish that on a mass spec. So they are good for analyzing. If you're analyzing thousands of things, you're usually going to be able to tell, 990 of those things apart. But there's going to be instances where you can't determine or distinguish two things as being different. So I just wanted to give you a very quick rundown of kind of what this data looks like. Again, you do some separation like on a LC. So each of these peaks is a compound that's coming out at a different time. So they're kind of like flowing along the column, and then they are annotated. This is where we figure out what mass they have, and that helps us figure out what compound it is. We then can do some analysis of comparison. So here we would actually say, here's my disease, here's my normal cells, which metabolites are different? Again, very similar to the comparison you would do in proteomics or transcriptomics. And in metabolomics, we also can take all the metabolites that we find and map them on pathways. You guys looked at some of these pathway analysis very, very, very early in the class. I had you look at some of the databases that give you pathways. Well, you could map each of the metabolites that changed in your disease cells, for example, and look at what pathways are they maybe related to. So again, this is some type of data just to give you a visualization. You could either look at individual metabolites and say, group 1 versus group 2 have different amounts, or you could then start looking at whole pathways that are changed. This is a plot of pathways that are impacted. So for example, in this particular subject, purine metabolism was very impacted. So is the glutathione metabolism. So its applications tend to be used for things like toxicity assessment, especially when you look at pharmaceutical companies. A lot of times they will want to test the toxicity of a potential drug. So they're going to see how the metabolites get altered in the organism when you give someone a drug. So this is a very common type of test, this metabolomic profiling. You have your, organism and then organism that has the drug, and you compare the levels of their metabolites to see if there's any toxic metabolites that are potentially building up, or any markers of problems that could happen from a drug candidate. Metabolomics also gets used to do biomarker identification. Just like in proteomics, you can do a comparison of disease or normal patients and see if there's a particular marker that might help you identify if patients have a particular disease. For example, they might have a hormone that's upregulated in their blood. Hormones, a lot of hormones are small molecules, so that would count under this metabolomics. Metabolomics also, because it is such a huge area of molecules you're looking at, gets used a lot for exploration too. So if you want to find something that is a unique chemical that's maybe used in signaling, or you want to understand something like the impact of disease or nutrition on your gut flora, that's just an example. You just want to explore what's happening. Or if you just want to, again, find unique components. For example, like I gave, you might take some plants and see if there are any type of chemicals that you've never seen before. And then you can pick those out and kind of look at what they do. But it would give you a way to explore new and interesting chemicals and small molecules that you didn't know about prior.